very effective team. On the left, we have Dr. Mohit Majumdar. Mohit is the Director of Business Development. He's a PhD in computational biology from Jawaharlal Nehru University, Delhi, a pretty prestigious university by its own repute. There is Pratim Chakrabarti, Pratim, who is me. I kind of look very, all those grave faces, but I'm kind of a little jovial person than what you can see in the picture. So I'm a PhD in bioinformatics, and I am the academic coordinator for the kind of programs that are being run right now across India. We have Shubhrajit Barua. Shubhrajit is a research intern with us, working for us for the last 2 months. Rishabh, could you mute yourself, please? Thank you. Shubhrajit happens to be the research intern with us. And he is working on some TCGA liver cancer project. He is in his BTech year, studying BTech in bioinformatics from Amity University, Calcutta. We have Apar Agrawal. Apar is also doing his graduation. He's just passed his courses in Amity University, Jaipur. Apar is taking care of some of the R programming principles, R programming stuff that are being used in our server platform for some basic machine learning data analysis. So this is our small team, and everything is led by Elia. Elia is here on the left. Elia manages all of us effectively. He's placed in Louisiana, USA. And this is our team from the Tauber Bioinformatics Research Center. This is our team in India. And we are kind of trying to bring this kind of cutting edge technology development programs and skill development programs and upskilling things to the audience in India, and so on and so forth. So we have, collaborations across the globe. We have collaboration with University of Nebraska Medical Center. Possibly you have been, if you visit our social media pages, you can see many of the programs that are being run right now in machine learning, in medical biotechnology, medical bioinformatics, and also specific sections like clinical genomics and all these sort, in collaboration with University of Nebraska Medical Center. There's another program coming up with UNMC recently, which would be ably led by Elia out there, who would be talking on some data science principles used in biomedical data analysis. Now, these are all translational things, translational courses, which are jointly conducted by Pine Biotech and University of Nebraska Medical Center. We have collaboration with Tulane. Tulane professors are a part of our program. They participate in our program, and they use our educational materials, educational resources, as well as our analysis platforms for doing their own research. Of course, we have University of Haifa. That is the place from where we have originated. We have Louisiana Biomedical Research Network, LBRN. In LBRN, there are various groups whom we work with to bring in value-based and project-based online, offline hybrid programs for the students of LBRN. And of course, these programs are also available across the globe for other audiences like you, who are willingly want to extend your learning, so that you have a better research opportunity, and also you have a better research, can translate your research to a different level, take it to a different level. We have Pennington Biomedical Research Center, LSU, that is also a part of Louisiana State University. This is also Loyola University, New Orleans. So similar programs run in these universities as well. And we have Georgetown with us. Georgetown is one of the leading, organizations in. Somebody is already having their microphone on, so I would request you, Mohit, can you mute that person so that we can have a,
Yes, Pratim, I've been muting people, but it's been.
Yeah, so I would request you, all of the people, to mute your microphone, because it creates a lot of disturbance, because you will be having those disturbances when you are listening to this lecture later on for your own understanding. So it's all for your own good. So please mute your microphone accordingly. Thank you very much. So Georgetown, as I was telling you, Georgetown is a very significant cancer research center. So, and we are in the process of, we are collaborating with them for analyzing the data, the big data that they have. And also they use our server platforms, which I'm going to introduce in a few minutes from now. And henceforth, we also have collaboration with Grambling University, Grambling State University, Bennett University, Times of India Group, University of Nebraska, University of Nebraska Omaha, and LSU Health New Orleans. So LSU has several sections, Pennington Biomedical Research Center, LSU Health New Orleans, and all these things. So they are connected with us for their own education and also for their own research. We have New York University with us. We are collaboratively developing translational programs with that. And I have already told you of our collaboration with Amity University. Now, Amity University, Kolkata has. So I would again request you to mute your microphone, please. Thank you. Yes, so we have Amity University, Kolkata. We have also have a few programs. We have a pilot running in Amity University, Kolkata for the last 3 months. We are in the second 1.5 months. We have traversed 1.5 months through. And we have a collaboration with CNCI. This is a local cancer center, CNCI, Chittaranjan National Cancer Institute, based in Kolkata, which is a very significant research and hospital. And they have very, very interesting research programs, and we are collaborating with them for analyzing their data, also accruing their data into data banks and databases, so that the researchers get a very good understanding about the various cancer dimensions that they have. And we are running some pilot programs with them as well. We also are running pilot program in Institute of Genetic Engineering. That's a private research institute again based in West Bengal. They are the first institute in West Bengal to start a biotechnology and a molecular biology course, besides Calcutta University and all those other private players right now available in the Indian soil. We have collaborated with Sister Nivedita University as a part of this program, and this was the ceremonial presence we have, presided by the registrar of the university and also the vice chancellor of the university, Dr. Professor Ashok Ranjan Thakur. He happens to be a very significant personality in the domain of molecular biology and bioinformatics in India. So, and there is the program, on that program particularly, so the program that we are running right now is a part of, employability enhancement program. And that's the whole reason why Sister Nivedita University is collaborating with us, because we also believe in the same principles. We have the same vision of bringing the right kind of translational education to the participants, to the students who are in the undergraduate level or maybe in the postgraduate level. The people who are undergraduate, they'll be kind of shown the path. This would be a lot of help to them to choose the right kind of career, to choose the right kind of upskill themselves in the right kind of process, so that they can be more employable and in the industry, and also in the research programs as well across India. The kind of skills, the kind of methods that are right now used are very much based on the high throughput technologies. So that's why, and also across the globe, the industrial requirement and the research requirements are very, very specific, and they require something else, something extra, which we study in our general curriculum. So the courses that Pine Biotech have, I guess many of you have visited our educational courses, educational platform, educational so that you can you have found probably a lot of courses out there. Maybe you have traversed to that, or else if you have not, you will get ample amount of chance in these 3 months to go through them. So in addition to our company Pine Biotech, some other company also collaborated with them on the same day. It was a grand ceremony out there. NASCOM people from NASCOM came over. Dr. Chintala, this is Dr. Chintala. She's the chairman of NASCOM. We have Data Science Foundation out here. Data Science Foundation happens to be a very significant institution which is trying to bridge the gap between the industry and the academia as well, so that people become more employable. We have Ticker Plant. Ticker Plant is based in West Bengal. They have multiple offices across India. They say so. They have some engineering. It's an engineering farm. They develop certain products which they want to teach those principles, those actually industrial level education to the SNU students. We e-litmus out there, and also Innovation Next. And the entire program was ably, presided over by Professor Anil D. Sahasrabuddhe, chairman of AICTE, All India Institute of Technical Education. I think most of you know this person, and this is the whole reason. I mean, this entire collaboration of employability enhancement was a guiding force, and that's we kind of match the vision of sustainability at the university, so that we can come up with this program called Data Science for Biologists. So yes, now I think we are in the process of telling you a little bits and pieces about the program that you're going to take up from today itself till November 23rd. So data science, as I was telling you, so we teach the principles of data science out here, and those principles are how they're actually used for the biological data. So our education is precisely the type of education which is, or the training that are required by the biologists, specifically the people who are into the domain of life sciences. May it be translational. We know that many people from startup companies are out here also, and how this program would help them. Really, they would help them to take the things actually how the things occur in the market, how to analyze the data. So we call it project-based online offline hybrid program. So because the reason why we call it online, because the entire thing takes place online. As you can see right now, most of you have joined via the online platform called Zoom. So this will be the link that will be used on every classes from today onwards till November 20th. We'll be covering the entire program is basically applications of data science in biology, biotechnology, and healthcare. So biology as in core biology, biotechnology as in on those technological advanced. Yes, could you mute yourself, Shomudeep? Thank you very much. So yes, the duration of the entire course is 3 months, and we call it workshops. The reason why we call it workshop, because every time in the class we give you the bare minimum necessary theoretical principles. We show you the bare minimum necessary theoretical principles, and also we tell you how to do it via using our server platform, which we call T-BioInfo. And I'm going to show you that a little demonstration of that platform as we go on. And our course is significantly divided into 2 parts. The first 2 months are extensive, the theoretical principles, and also the actually hands-on learning, where you use the platform to analyze the data. You understand how the data are analyzed. You understand about the various data science algorithms, the statistical principles that are typically used to analyze your biological or biomedical data. And on the last month, there's a project. So what is the importance of the project? the project is actually the culmination of the learning. So either you design your own project based on the things that you learn, or you can use a number of research projects that you have, that we have in our educational platform. So all these things, I'm just telling you, but I'll be showing you as well as we go on. So 3 months, 13 workshops, and one team project. And we will be using something called T-Bio platform. And definitely, these classes will be taking place on Tuesdays and Thursdays from 8 p.m. to 9 p.m. So twice a week class for the next 2 months, to be very specific. And the last month is totally dedicated to the project. And at the end of the project, we are planning to, we have a workshop again, where you display your your work as a poster, or as a small session, something like that. I mean, so it all depends on the kind of things that you do. So there are two options: either you choose the projects that we already have in our educational site, or you kind of if you are trying to solve some problem on your own, some biological problem on your own, you can do it also in the last month research project, which is going to take place towards the end. So the program will be guided by two people, myself, Pratim Chakravarti, and I'm a PhD in bioinformatics and data processing, and also Dr. Mohit Mazumdar. Mohit is a PhD in computational biology and machine learning from JNU, as I have already told you. And reinstating the fact, online classes on Tuesdays and Thursdays from 8 p.m. to 9 p.m. And be very sure these classes are kind of going to give you the right breadcrumbs to go along. So what do I mean by breadcrumbs is that these are the pointers. So we definitely we are expert enough to tell you where to start from. So data science is a domain which is comparatively new as a jargon, but data science was there in the form of statistics and computer science and mathematics for a long time. Now we have coalesced all of them together in a principle called data science. And we teach you the right kind of algorithms which are used for analyzing the data. So this is how a little bit of introduction about myself. I have been working in the industry for almost the last 15 years. I have worked in software companies. I have worked in academia, working as an assistant professor. And then I went back to the industry and worked in concerns of I was based in Hyderabad for a long time, working for a prestigious contract research organization out there. And I've also worked on some other software companies who have life science verticals. So, and thereafter, my experience in this particular domain have landed me up a collaboration with Pine Biotech. And I'm in front of you to give you the right kind of assistance and also the right kind of abilities to upskill yourself, and so that you learn the essentials of data science. So there is no end to learning, as I can tell you, but there is always somewhere to start. So maybe who are here, they may have a lot of bits and pieces of knowledge here and there. I mean, I can tell you my experience that, and if you call myself, do I call myself a data scientist? No, I don't call myself a data scientist. I understand what is data science, and I know how to use the data science in the biological domain. And the kind of things that I know, I can share with you. I can share the knowledge with you, and maybe it would come to your help, and maybe you can become what you are trying to look at yourself in the coming years. You can become one of them if you start really doing the kind of things that we show you, or we kind of guide you to do. So we understand what is data science. We understand how these data science principles are used in biology, and also the life science data, to be very specific, starting from biology data to biological data to biomedical data to healthcare data. All everywhere, there is a lot of usage of data science here and there. But again, what sort of principles, what sort of algorithms, what sort of methodologies are used? These courses, the course that you have participated very carefully, would tell you to give you that particular guidance. And to tell you about Mohit has been there in academia for a pretty long time. He has substantial experience of teaching, and he has substantial experience working in the industry as a business development person. He understands the science very well. He understands the data science also very well. Being a person from machine learning, he understands all those machine learning principles, which are typically, that has been there. I mean, machine learning has been there for a long time, but again, same thing, the data which typically were used. So by virtue of all those upcoming technologies like high throughput data generation, so there's a huge deluge of data in the biomedical and biological and life science domain, and thereafter machine learning comes into play. So now it's a right time to, if you want to start your career in data science, maybe this is the right place, and maybe this is the right time as well, so that you can reap the right kind of benefits in future years. Okay, moving on. So today is the first session. We call it a workshop because it is an interactive session. As you can see that this is an interactive session. So anybody who wants to ask a question can raise their hand and ask a question right here. So I would take a few seconds of pause, and I would ask if anybody have any questions. They can unmute their microphone and ask me. Also, you can write in the chat as well, so that I can answer your question as well. Any questions you have? So, Sunita, I'm audible. Am I audible clearly now? Anybody? Any questions? You can post your question in the chat as well.
Hello.
Hi,
Actually,
Christian.
I have a question because
Please.
some technical problems. I couldn't hear the part of the project.
Okay.
I don't
Yes.
know if you can repeat
Absolutely.
that part, please.
Absolutely. Christian, could you introduce yourself, please? That would be very nice, so that all of us know each other.
Oh, yes, of course.
Thank
My
you.
name is Christian Guadarrama, and I'm from Mexico. Right now, I'm a master's degree student, and well, I'm doing a project about cervical cancers and the application of biotechnology and PCR protocols to detect some specific species of lactobacillus in cervical exudates.
Amazing. Thank you, Christian. Yes, nice to have you here in this program. I think that it would be very helpful for you to use the data science and all these things. So, Christian, I would be coming to those specific project related stuff as we go on. Now we are in the process of explaining all the programs day by each day. So I'll be describing more about the projects that I talked about a few minutes back. Thank
Okay,
you. Yeah.
thank you.
Thank you, Christian. Anybody else? Any questions you have? So we have people from South America. I think we have people from India. We have people from Africa as well. Hyacinth, I think, is also has joined us. So Hyacinth, if you have any question, or anybody else, any questions you have, you can write in the chat as well, so that we can address it. So I guess there is no question right now. So I will take the liberty to move on. So today is the first session where we are going to introduce the program, and we have already introduced the program trainers as well. So I'll be giving you uh, the broad overview of the program resources that we're going to use. And also I would be telling you about the training program curriculum that we are going to cover each day, and what are the things that will be covered here, and also the important deadlines that we have in this program. So again, the course that you can see right now in front of yours, we call it Omics Logic, because we teach the logic. We kind of connect the omics data, multi-omics data, from genomics, proteomics, metabolomics, metagenomics, and epigenomics by the principles of logic. And by logic, what we want to mean is basically the machine learning principles that we use. We are going to tell you in this particular course. So that's why we call it Omics Logic. And this is also a part of the Omics Logic program. We have named it Data Science for Biologists. And today is August 20th. So I have told you about the introduction. I have told you about the trainer. Now let us go to the resources which you are going to use. Probably you can see my screen. This says T-Bio platform, T-BioInfo platform, user-friendly platform for bioinformatics and machine learning. So pretty soon, you will be receiving an email with a user ID and a password. So what would be your user ID? Your user ID would be the email by which you have registered into the program. So do not worry. Just listen to me very carefully so that you don't have any confusion at the end. But always we are happy to help you via various methods, via Facebook, via LinkedIn, via all those social media platforms, and all that. At the same time, we have emails. So we call our courses basically advanced MOOC, massively open online courses. So why do we call it advanced MOOC? The reason being that we have a very significant feedback mechanism system. So in gene expression networks, there's a very significant feedback mechanism system which actually tells you when to start and when to stop, and what to expect and what not to expect. So we kind of reach out to those people. So there will be pre-assessment to this program. There will be post mid-assessment to each of the modules that you complete, and obviously at the end of the entire program there will be post-assessment. So in the pre-assessment, we try to understand at which level the participants are. For example, we can see many people coming from different backgrounds. For example, Christian said he's trying to do a project on cervical cancer, and he's trying to understand some of the biological pathways, maybe by using the biological data that he has, and also using the machine learning principles that he's probably interested in right now to understand from this program. So coming back, so feel free to ask questions, to post the question in the chat as well, and when I give the next pause, we'll be happy to answer your questions accordingly. So this is the server platform that we will be using. Why we call it project-based? We call it project-based because we give you the data on the beginning and the first day of the program, and we teach you the basic principle, the bare minimum necessities to swim around in the ocean of data science, and we give you this platform which you can see right now. The URL of this server is https://server.t-bio.info. So you have not received your credentials as yet to log on into the server. You will be receiving that soon. For the entire span of 3 months, we will be you'll be having the access to this server platform, and you can do all sorts of data analysis, primarily in the domain of NGS, mass spectroscopy and structural biology, and data integration. Now let us go to the actual server where we can tell you about the thing that is there. So again, this is the server platform I have. You can see my screen. So I have already logged on. So what would happen here is basically when you log on, once you log on, the screen that would be shown to you, let me open a new window so that I can show it to you. So in the meantime, the browser opens. I would be happy to answer any of your questions, if any. Okay, so the address is server.t-bio.info. So probably my browser has already recorded my input, so that, yes, I'm already logged on here. So I have to log out probably. So signed out successfully. So again, we do a log on. So since my browser has remembered the password, so this would be my login ID. See, this is my login ID. So whatever login ID you have given us to register you with will be your login ID. and the password will be sent by us. The password would be something which we would generate for you, and we would send mail to each of you individually, so that you will have an account of yourself in the server space. So this is based out of cloud. So let us log in now and find out. So let's do a login. So once you log in, something of this sort, something of a message of this sort will be shown, signed in successfully. And this is a server which has been developed by Tauber Bioinformatics Research Center. Okay, for almost a decade of research has gone into it to develop various pipelines and various algorithms. So this is a seamless UI. We call it a seamless semantically connected UI, user interface, where a number of algorithms from various domains, primarily NGS and mass spectroscopy and structural biology and machine learning things are used. So the algorithms which are connected to NGS data analysis are there in this block. The algorithms which are connected to mass spectrometry are out here. We have a small structural biology, set of algorithms out here, which primarily uses the data from the PDB. So the input to these particular pipelines are basically the protein data bank structural files. Please take note that this is not a place to do your simulation from. So computer simulation, molecular dynamics simulation, that is beyond the scope of any of these cloud-based things. So probably you want to use some other set of installations like Charm and Amber and GROMACS for this thing. But before actually doing the simulation, there are a lot of data science which comes into play to give you the right direction of your data analysis. So these are some of the principles that are used, And we have a specific set called virology, which are used by University of California, Santa Cruz department. We have a collaboration with them. So we have a small virology team out there who wants to use many of the principles that are there in our, many of the pipelines that we already have in our pipeline. And this is the data mining and the data association part. Most of the, our chief, USP of our platform is multi-omics. I will come to that one by one. So we have all these things, and we have the data. In the data mining part, we covered the supervised analysis and unsupervised analysis. And this part is basically the machine learning part that we will be covering in most of our, in the entire length of our courses. So we'll tell you about the basic principles of the domains that we are going to use, and we'll be telling you how to actually use various types of machine learning principles to analyze those data. Now let us go around a little bit, play around a little bit, so that you can also play around a little bit once you have the server login. But there are rules of the game, I can tell you. So there are specific rules to the game. You cannot just upload any random file in out here. You cannot just randomly go around this platform and try to run unnecessary pipelines. So, but again, running unnecessary pipelines will not really give you the right kind of result. So that's why we have created small pockets of user-friendly pockets, which we call it demo. Now let's see this. We have our demo RNA-seq cell line. We'll be covering this in the transcriptomics part. Let me show you a small demonstration of that. So we will be dealing with these demo pipelines a lot during our course. So as you can see that typical RNA-seq analysis consists of many portions. And so this platform, we call it, it's helpful for both newbies as well as all those people who are kind of experienced. Like, there might be experienced bioinformaticians out here who want to find all the algorithms here. So this platform is basically something which is very similar to Galaxy, but something which is a little more advanced than Galaxy, or the 7 bridges that are there in the right now available in the market. And this comes from a very significant research group, and we are continuously in the process of development of this particular platform so that it becomes more able and more solid for analysis of your data. So yes, let us go back to the demo pipeline a little bit. So these are the various steps of pre-processing, data simulation, error correction. Please do not, feel free. I mean, please do not feel hesitant about going into this demo pipeline once you have the have your login and ID password, so that you can get a feel and get a upstart about a head start about the courses that we are going to conduct in future in this as a part of this course. So mapping onto the genome, mapping on junctions, isoform construction, gene transfer format processing, mapping of transcripts, ROC mapping, expression levels, differential expression, segmentation, and obviously all those annotation, which is the very important part of any sort of analysis. So for example, I can show you one of the pipelines that I've already run. For example, these are some of the pipelines that I've already run. I have run some pipeline on factor regression, and I have run some pipelines on quantile normalization and some bow tie RSM table of expressions. So these are some of the name of the files that name of the pipeline runs. So it shows me this is the entire pipeline listing, number of pipelines that you have run. I have run, what you can see that I have run all these pipelines for the last few months. You will also be running the same thing at the, as you go on into the course. So this is how the server looks like. Now, very interesting about this platform is that if you hover around in these particular blocks, for example, NGS data, it tells you about the NGS data, that what is actually next generation sequencing in a nutshell. So anybody who is a newbie, who is a biologist by training, he or she wants to jump into the world of next generation sequencing. This is the perfect place to start from. So it tells you about the NGS data, that what it does, and thereafter lists all the basic principles or the basic broad analysis areas which actually are there in NGS data analysis. So each of these blocks have have all these pop-ups. We call these educational pop-ups to guide the initiated user, or to make the user more initiated about the typical analysis principles that are used, and so on and so forth. Okay, and now let us go back to the presentation a little bit. This is the the UI that you see once you log on. I already showed you. I can go back to that later on, might be, because we are kind of running out of time. So let's go on accordingly. So these three are the basic formats which are used in our platform: FASTQ, FASTA, and GTF, gene transfer format. So again, FASTQ, FASTA, these are all some of the formats which are the output of the next generation sequencing platforms, be it Illumina, be it any other company, PacBio or Oxford Nanopore, etc. So these are the inputs to the pipeline of algorithm that we have. And the blocks, these color blocks actually shows you that how the server works. So it starts from pre-processing, it starts and then it moves on to mapping, and then it moves on to the quantification of the expression levels and differential gene expression. So these 4 blocks, actually, if you go from top left to the top right, and then to the bottom left to the bottom right, this is the flow: top left to top right, and from the top right to the bottom left, and then to the bottom right. So for a typical gene expression, gene expression determination, these are the principles that we are typically use. We start with pre-processing, mapping. So this is a very sample, very broad overview of. It would make more sense as we go on into the course and tell you more about the pre-processing steps, what kind of things are actually the input of those pre-processing steps, what are the sort of output that we typically expect in these mapping and quantification of expression levels. So this is how a typical pipeline looks like when you are not running a demo. Okay, so you are not running a demo. So this is how it looks like. So I would show you in the server how it actually looks like. So we call it, as I was telling, user-friendly platform for bioinformatics and machine learning. So let us go to the server and find out what kind of, where do we really encounter this sort of UI. Okay, I click on the transcriptomics, not the arrow. So let me go back and show you once again. So in this place, if you can see my screen, probably you can find something called RNA-seq chip parallel analysis of NGS and microarray data. Now, beside this, there is a small arrow, small down arrow. If you click on the down arrow, you would find demo RNA-seq cell line project and demo RNA-seq Angelman project. Now, you want to analyze your own data, let us say. You do not need a demo. You need something, some working pipeline. So what you do is that you go in the RNA-seq chip parallel analysis of NGS and microarray data. You click on that. You have a UI like this. Now, just compare the UI. See, this is same. You have all those buttons that you can see in this figure out here also. So these are the dashboard buttons. These are all the running pipelines. I showed you some of my pipelines. We have a specific pipeline, create new pipeline tab, which you can find out here. And we have all those options that we can find. So advanced option, predefined options. So the UI which is shown out here is, this is the latest UI that we have. We have separated it at preliminary and file upload, two primary steps. So here you can have all those reference genome that are there in our server. You can use the data that is present in our server. So or you can, when you are doing a bioinformatics project, you can access the something. You can also upload your own data and do it. So we'll come to those details later on as we go on. Now, the format of the NGS data, the typical text space, I was telling you that FASTQ, FASTA, SAMBAM. SAMBAM is actually the extended format of GDF, and we have simulate FEQ and simulate FQ and simulate FA. Now, typically in all the NGS experiments, there are single end reads and paired end reads. These two are connected with the coverage and the depth of NGS analysis. So we have all these options available out here, and the set of organisms that are there are these Arabidopsis, all those standard model organisms which are used in biological data analysis are there. Arabidopsis, Homo sapiens. So we have two distributions of the human genome. One is distribution GRCH37 and GRCH38. Musculus, Rattus, and Drosophila, Canis, Saccharomyces cerevisiae, and Aedes, Bos taurus, Dani, and bacteria on all these things, human mice concatenated. And this is the pipeline that we would be building in due course of time. So, but this wouldn't make sense to you really. So whatever you see in the demo part is actually starts from here. So what is the significance of the demo is that, this is the slide that tells you about the demo pipeline, that where you can find the demo. So what is the difference between demo pipeline and actual pipeline? First of all, the demo pipeline has already been done for your usage. So all those files which has been used as an input has already been done. We have already made that effort on our part so that your life becomes a little easier. So this is kind of a, you can go through it. So it's a mock pipeline that we have done, but actually it does some analysis. We have done that analysis. We have stored the file out there. We have recorded the entire pipeline in some form or the other. We have used some technology out there, and we have given you the necessary output. So if you run the demo pipeline, you can find at the end of the day the results. So let us run the demo pipeline and show it to you. Okay, so that would kind of maybe demystify about the demonstration that are there. Okay, now let us learn this demo RNA-seq pipeline. So we start. So see, in this entire set of buttons, so all these are algorithms. The algorithms which does not have this small T-shaped symbol are, so the algorithms which have these small T-shaped symbols are actually those symbols are actually those algorithms which has been developed in Tauber Bioinformatics Center. So these are proprietary algorithms. So our platform has a very, balanced mixture of proprietary algorithms as well as open source algorithms. So Coral, Eco, RESA, these are all open source algorithms. Clean Primer, Array to FASTA, for example, Array to FASTA is a program, is a algorithm developed in Tauber. So again, RSEM, SynR, Top Hat 2, HiSat 2, Bowtie 2T, Bowtie 2I, and all these things are kind of available in open source, maybe mostly available in the bioconductor as bioconductor packages, BlastX, and all these things. So these are blocks, pre-processing, followed by pre-processing, there is error correction, followed by error correction, there is this exon detection and mapping onto junctions and mapping onto the genome. And these are the set of algorithms. So if I click on in the entire set of button, the only button that is right now highlighted is start. Now let's click on that. This is an AI guided platform. So which actually tells you, which guides you, what are the best possible options that you can use to analyze your RNA-seq data. So the button that is there right now highlighted is start. Now let us click on start. So this is what we call the educational pop-up. Okay, if you scroll down, you can find what is this pipeline that is going to be built. Okay, and you can find these boxes. I showed you these boxes a few minutes back: pre-processing, mapping, and quantification of the expression levels. This particular educational pop-up is meant for people like you who actually are newbies in this platform. So once you become an expert, you won't be needing all these pop-ups. But again, you cannot just bypass this thing, because there are, in many of the pipelines, there are a number of algorithms which might require some help at the end of the day, so that instead of just going frantically finding some reference point, it's better to keep the reference points at one place so that you can go around and find what you are actually doing. Now let us what is the next step? The next step is basically so let's look at the input files. What are the input files? The input files are samples.svl. SVL is a soft link to the main big data of the data that are already uploaded in the server. And there is something called cell lines clinical type.txt. The output files, there is no output file as of now. So we have only the input file. These two are the input files. We'll learn about all these input files as we go on in our course. So let us click OK. Now the immediate next step is in the same box. What is the next immediate box that is being highlighted? Trigonometric. So the platform tells you what is the next immediate step. So trigonometric is the next immediate step. So what does trigonometric do? Trigonometric actually maps onto the aligned regions and try to align the on the reads onto itself. And there is some interesting publication that are attached to it. So these publications, you can click on these publications and you can open those publications and read about all these things. As I was telling you, that we always in this entire length will be giving you breadcrumbs to chew upon, to actually understand the process of the actual data analysis. So the immediate next step is trigonometric. We again here also, what is the input file? The input file is already there, uploaded in the start. So this is the first step. So we click on OK. See, this connection is built. So from start, the input file was sample.svl and also clinical samples.txt. And the first algorithm that we have chosen is trigonometric. The very immediate button that is being highlighted right now is PCR clean. So we have to click PCR clean out here. So what does PCR clean does? This entire explanation is there. What does PCR clean do? And also the basic steps of library preparation and sequencing are explained very clearly out here. And also there are hyperlinks out here which you can study and find out what actually PCR clean does in the process. So what is the input file? You can say that input file already been given. we are trying to build the pipeline. So input file has already been uploaded in the beginning of the step. And as usual, there is no output file out here. Yes, Abhishek, you have some question?
No, sir.
Can you just mute yourself, please? Thank you very much. Yeah. So the very immediate step which is being highlighted in the next box is HiSat2. Now HiSat2 is an algorithm which actually maps into the genome. Let's click on it. Okay. See, this is the again another educational pop-up that has come up. So this has also, all these algorithms are connected to publications. All those highly cited and highly respected publications are from there. We have collected all those algorithms together. That's why it took a lot of time to really analyze and integrate all these principles in one place. So you can reiterate. I mean, what does HiSat2 does? So now we have started, and PCR chemomatic, PCR clean, HiSat2. What is the next immediate step? That's isoform constructions. And what is the algorithm that is being connected right now? So as I was telling you, this is a demo pipeline. So this is one of the bare minimum basic step of any sort of RNA-seq data analysis. So click on cufflinks. So cufflinks is the next immediate algorithm. So what does cufflink does? Same thing, some educational pop-up which explains all the necessary things that goes into the, what does actually cufflink do, and algorithm does. And we click on that. So probably you can right now find the meaning of the colors that I was showing you. Pre-processing is blue. See, the pre-processing is blue, right? So the mapping is basically red. So mass mapping, HiSat2, this is red, right? Isoform constructions, this particular pipeline, the color code that you see right now, does not take isoform constructions into consideration. So that this is a differential gene expression by box that we are showing you. So next immediate step out here is cufflinks. And what is the next immediate button that is being highlighted? Cuff merge. So let us click on that, GTF processing. So again, the same another educational pop-up comes up. It tells you about the publication that will definitely explain to you about the this particular algorithm. You click OK. So here we are building the pipeline, the input remaining the same. The immediate next button that is being, so cuff merge, as you can see, is also mapping, right? So here is one mapping. We try to, we do the isoform construction, and we process the GTF, and we use cuff merge for that. And then we go to the mapping onto the transcript, which is bowtie 2T. Okay, so again, the same thing, the bowtie 2T here explains that what is bowtie 2T and the associated publications, publication that has been, from which this concept has been taken from. So this is also a part of mapping procedure. What is the next immediate step? The next immediate step is RSEM table of expressions. If you can see my screen, the expression levels are shown in green out here. So we have completed all the steps of pre-processing. We have completed all the steps of mapping. And now we have come to a stage where we want to quantify. We quantifying the steps here, quantifying the expression levels here. Okay, so yes, now let us click on RSEM table of expression. And RSEM table of expression will tell you that what is the output would be like, what would it look like, and also the associated publication, why this output and all this stuff. Please go through it once you get your server credentials available. And then we have come to the level where quantification of expression levels has been done already. What is the next level of next button that is being highlighted immediately? What is differential gene expression? Differential expression, DE-Sec2. Now the educational pop-up, it tells you what is DE-Sec2 and the reference publication attached. So you click OK. So once you click OK, that particular pipeline is selected. So if you want to unselect that pipeline, you can right click on it. Okay, all this time for actually selecting that particular pipeline, I was clicking on the left mouse button. If you want to unselect it, the button that you should press is actually right mouse button. So the pop-up comes up, a drop-down menu says, server.t-bio.info says, are you sure you want to jump to the previous step? If you click OK, you would be coming back here. Okay, now yes, there is some problem here. I think we need to solve it. Anyway, the immediate next step that is being highlighted is end. Now let us end here, and the entire pipeline is built. So this is the pipeline that I have built right now. We have started with inputting with input file some SVL soft link file, and also the clinical some textual file which contains the necessary information. We have gone to Trimomatic, PCR clean, HiSat2, cufflinks, cuff merge, bowtie 2T, RSEM table of expressions, and DESEC2 and end. So as you can see, this is very easy to build. So I can tell you a lot of work has gone into it to make these look so simple, because you know the kind of things that you really require to figure out, to semantically connect each of the algorithm with each other is huge, because you know you have to understand some formats, you have to understand the dependencies. So here you have to, we are making your life much simpler by taking all your headaches about the computation infrastructure, and also trying to understand all those semantic connections between a number of algorithms that are there in this platform. So that's why we call it Smart UI, seamlessly connected and semantically connected user interface for doing your bioinformatics analysis. Going back to the presentation again, so let us quickly finish it. So I told you about what is the significance of demo pipeline. We have another platform called edu.t-bio.info. This is the educational platform we have. So let us go to the educational platform quickly and tell you about this. I think all of you have been able to log on. See, this is my, I have already logged on, so I can show you. So these are the, you know, the breadcrumbs that we are giving you in this particular course, and many of the courses that are coming at the same time. So the address is edu.t-bio.info. I would paste that in the chat so that you can see it on your own. So I can see many questions here. Yes, Apar, Shomudeep, I can't access. So yeah, Shomudeep, you won't be able to access the server as I was telling you, because you don't have the credentials. We have not given you the credentials as yet. So Naresh, to everyone, please zoom out the slide. I don't understand this. Okay, yes, Apar, and yes. Naresh, if possible, zoom this slide, which slide you're talking about, to be very specific.
Pratim, hi Pratim, hi.
Yeah.
Hi Pratim, Mohit this side.
Yeah.
Yeah, so this is the book. We showed you the platform, and the platform is the experimental table where we are performing the experiments. And this is the detailed book that will give you the insights, the things that we talked about. So everything is explained very well in here so that you can actually read through it and perform all those analyses. And we'll have a small demonstration about the platform. So I think we have already exceeded time. So that's one limitation.
Yeah, I would take a few minutes just to go through the educational platform so that they can have a quick overview.
So here, as you can see, there are multiple ways to, you know, log in into the platform. You can use your Gmail account, you can use your Facebook account and things like that. So anyone can use the platform, the educational platform. Many of the courses are free, but many of the courses requires the access to the analytical server.
Yeah, so I have logged on into the number of courses here. You know, if you go to the course collections, you can You can see a number of courses out here, for example, bioinformatics, epigenetics, machine learning, metagenomics, genomics, and transcriptomics. And let us go to the presentation and quickly finish it. So you visit the server accordingly. These are the courses, how it look like. And you get certificates at each as you go on doing it. So this particular course is, and at the end of completion of this course, you would get a certificate from Pine Biotech and Tauber Bioinformatics Center, which is internationally valid. And each of the courses that you cover in the educational platform has certificates attached to it. So if you complete machine learning, you'll get a certificate of your own, which you can portray in your CV, you can portray in your social platform, and so on and so forth. And I quickly go through the forum. That is very important. So let me copy this. So this is the forum where you can find many things out here. Sorry. Yes. So this is the forum which you would be able to access. So this is the forum. Here you can find the curriculum out here, what all we'll be covering. So I have not registered, that's why it's not showing me anything. So please register out here so that you can go on. Okay, so I think there is some problem with my.
No, Pratim, you have to log in. So you're not logged in.
Oh,
That's
no,
the reason.
I'm
Yeah.
sorry. Yes. So let us go from here.
So this platform is limited to the paid participants. They will have a forum here, which Pratim will show you after a while.
So there are a lot of securities associated, so it will take some few minutes of time. Let us see if I can log on now. Oops. I don't know what's happening. Let me go here. I think I have logged on here so that I can. Okay, so this is the curriculum. Let us go to the progress bar. And we click on the curriculum button. This is what we are going to cover. And there's something called forum here. And here you can find all those previous presentations, and today's presentation and link to join today's session has been posted already out here. So I would tell you more about this thing. So one task you have when you have, you go to this page and register yourself. And one assignment that you have, that you complete your profile. For example, I have my profile out here. If you go here, this is my profile. So if you go to your profile, you upload a photo of yourself and also write all about yourself, so that we are also trying to create a community here of like-minded people, and post your, this is my photo, and also you kind of, add some something about yourself, that why kind of what actually you do. For example, the way Christian said about himself, and this is what my detail is. And you can add your Facebook page and Twitter page so that you can directly share your credentials, which you gain out here to the rest of the world. Okay, and quickly going through the dates. So you'll be having this presentation with yourself, so that the next session is on 22nd August, where we'll be talking about the knowledge-driven and data-driven discovery and examples, all those applications connected to bioinformatics and agriculture, neuroscience, etc. The central dogma, the basics of molecular biology, also the mutational variants, the genome variations, and specifically about targeted whole exome and whole genome sequencing, and the algorithms which are connected to genomic data analysis. On August 29th, so twice a week class, hourly class, 8 p.m. to 9 p.m. Next generation sequencing transcriptomics, all those raw NGS data reads, how to analyze and quantify them, interpretation, and going into transcriptomics 2, filtering and exploring and principal component analysis, the basic, here we introduce the first machine learning thing that are used in analyzing the data. And we have the second workshop. The first workshop is today, where we explain the program to you. The second workshop we are going to organize in September 10th. The time will be communicated to you. Probably it will be in the same time from 8 p.m. to 9 p.m. And we'll be communicating exactly the thing. But ideally, it should be in the same frame of the time. Immediately followed by the workshop, we have Introduction to Metagenomics, which is on September 2017. This is something, a very new type of study, which we are bringing in this is a comparatively new discipline that has really come up into the market. And we cover Metagenomics 1 accordingly. We understand how to logically analyze the metagenomic data and methods and interpretation, and definitely visualization of the results. And on October 17th, we have the Introduction to Machine Learning, and supervised and unsupervised machine learning, followed by the Machine Learning 1, where we understand about decision trees, discriminant analysis, support vector machines, etc. And on October 24th, we kind of complete the theoretical part of it, where we also do a lot of hands-on as we go on doing the transcriptomics, metagenomics, and genomics, etc. We'll be covering a lot of hands-on thing using the server that I showed you a few minutes back, for which you'll be receiving the credentials. So planning the bioinformatics project, this is what I was talking about, Christian, that either you can plan your own bioinformatics project for a month-long project with the kind of data that you want to look at. We would typically we look at team projects. The team projects is like the one which take. So if we have a substantial number of people, maybe instead of all the people doing individual projects, we can team you people up, and we can help you to identify the correct data sets and frame a bioinformatics project of your own. So we already have a few projects in our edu platform. Feel free to go around in the edu platform and find out what all are there. So we have specific projects on cancer, on exosomes, and all those domains. And we have a number of projects here. And this is what your project, this is the place where you can create your own research projects. So we call it practical assignments and theoretical curriculum. We have a lot of the bare minimum theoretical thing, hands-on and independent thinking. And totally, we try to give you a research experience, which actually is needed to get you a job in the research domain, as well as in the domain of industry as well. So the workshop 3 that will be taking place in the October 25th, which is actually designing the project proposal for yourself. That what kind of project do you want to do? What kind of data sets do you want to choose? What kind of data sets? Where can you find those data sets, etc., etc. And as I was telling you, at the end of this entire course, we intend to have a conference where individual participants or a group of participants will present a poster of their own. We'll organize a day-long or a 2-day-long conference where we will do a lot of activities from on the current nascent areas of bioinformatics, which covers the data science principles, are huge use of data science principles. Now, this is some of the workshop that already happened. This is what we typically intend to organize, the bioinformatics conference. Dates are about to get announced as we go on. So we select specific publication from which curate the data set, and we this is one of the session which happened at LBRN, Louisiana Biomedical Research Network, where we did this kind of program earlier. These are some of the feedbacks that we have got of the programs that we have done till date. Dr. Ian Townley is actually a professor of practice at Tulane University. He is a part of our precision oncology program, which I'm going to show you. Vok has been there with us for a very long time. She's a molecular biologist by practice, but she has taken our courses and already started using the bioinformatic principles in her own research. She's based in Nigeria, so probably Hyacinth, you can get in touch with her to know more about the course that we run typically. So the programs that are being run right now across the globe is bioinformatics for precision oncology. This is almost going to end in August 30th. We just started in June 10th, where we covered all those basic bioinformatics principles in the domains of genomics, transcriptomics, and epigenomics, and all those data resources which goes into precision oncology program. This is I was telling you about another program, Introduction to Bioinformatics, which I've started from July 19th, and it's going to continue till October 19th. This program is also going on with us. This is another program which is being headed by Elia Brodsky and Despina and Sahil and me, Omics Logic Genomic Data Analysis, which has already started. We are almost midway into the entire program. This takes place in, again, a same online program that is there. And this is another program that is going to come up with us on 3rd of September, the collaboration with Institute of Genetic Engineering and Data Science for Biologists. The same thing, if anybody does, I mean, ideally the same thing. So anybody who would like to kind of have some time constraints, they can join the program, which is going to start in this part of the, on September 3rd. Thank you for a patient listening, and I would try and give the stage to Dr. Mohit Mojumdar if he wants to add something to the entire presentation.
Yes, Pratim, thank you so much. This was really wonderful. And to add a few things, the IG program will be happening at a daytime, so probably around 1 to 2, or I guess 12 to 1 in the morning. So yeah, and now we have a session for question and answer. So anyone who has any question, I see Arpan Das has raised hand, Arga Datta has raised a hand. So if you guys can, unmute yourself and ask us. So yeah, I see Soumyadeep Mandal.
Will anything happen in our campus regarding this course?
Yes, Soumyadeep.
Yeah.
Actually, in SNU there will be 2 workshops that will be on-site workshops, and everyone who has paid for the course and is in the same locality can come and join us there. So the workshop will be happening in the daytime, but the time is not decided, but the dates are already shared with everyone through our landing page, and the dates that we have already shown you during the presentation.
Hello,
Yeah,
Mohit.
Argo wants to know the payment. Yes, please go on.
Yes, I have a question about the
Yes,
class.
please. Yeah, sure.
Are they taking place in the same platform? I mean,
Yes.
in Zoom platform or in other platform?
Which one you are trying to say? Once again, sorry.
Yes, if the classes are taking place in the same platform.
Yes,
I mean,
it will be taking place in the way you have logged on today. It will be done in the similar way. You can unmute yourself, ask questions in the chat, and we'll be using the server that I showed you entirely throughout the course.
And then you'll be practicing on the server.
Yes, we'll be given the user ID password very soon. So somebody has already asked that approximately when do we get our login and ID and password? You will be getting the login and the ID of the password by the next class. So the immediate next class is actually the big data. We enter into the technicalities of the thing. So you will get all the details out there. So Argo, if you have registered, if you have paid us, you would definitely get the login and password.
Okay, for example, when we get into the BioInfo platform, we'll simultaneously have classes at Zoom and we'll checking out the BioInfo platform as well?
Yeah,
Yes.
absolutely.
Yes.
Yeah, that's how it's
Okay.
going to be. So there will be practical sessions, there will be hands-on throughout the course.
Okay, excellent. Thank you.
Yeah. So we show you the platform. Then you log on into the platform on your own. We in the class also, we kind of give you that feel. You can log on at your end, I can log on at my end, all the participants can log on simultaneously from their computers. Christian.
Okay, thank you so much.
Thank you so much, Christian. Good to have you here. Om Prakash, kindly provide enough notes before one day for coming lectures. Yeah, we'll be giving you what kind of things do you need? Notes, written notes?
Prakash, actually, we have the courses for that. So once we will go to the next session, we'll make you understand. So I don't think you have to be worried about the notes.
So Sylvia Vasquez wants to know that he wants to join the course. She wants to know the cost of the course in dollars. So Sylvia, we have your email, so we can send you the details accordingly. So she's interested in database for viruses. So we can have a chat over email, maybe.
Yeah, we will reach to that point during our last
yes.
session,
So
and we'll discuss more about the projects.
Correct.
As Pratim has showed you the process that would take you to start a project. We'll discuss all those things during the upcoming presentations and the classes.
Correct.
Yeah.
So all those things that specifically people are looking for, working with viruses, working with bacteria, working with metagenomes, all that can be done provided you know the entire operations of the platform. That is what we are going to teach you. That's what we call it hands-on practicing online training program. So it is all about this only. Thank you, Sylvia.
Yeah. Another thing I want to add here, that if you are a student of SNU, Sister Nivedita University, then you have to pay at the university.
Mm-hmm.
Yeah.
So Shomudip says, I'm not a biology student actually, but I'm interested in ML and application of ML in biomedical. Yes, you do not have to worry. We will teach you, we will give you enough assistance so that you understand all about the data. So for you, if you are I think you are a computer science guy. So for you, data is all about, bits and bytes. So all characters. So don't worry about that. Don't worry about that. I guess I think we met you at one of the induction workshops. So thank you. That's not to worry about. You have to. Arpon, yeah, thank you. Arpon, if you have joined. So any more questions? Om Prakash, I think you got your answer. Shomudip has got your answer. Om Prakash, yes, 1 hour is less. Mohit, thank you very much for, yes. Yeah, thank you. So 1 hour, typically we cover a lot of time. 1.5 hour time we typically try to cover. Orgo wants to pay so that, I do not know where you come from. Orgo, are you from SNU? If you're from SNU, you can directly get in touch with one of your professors, or you can get in touch with us via email.
Soumyadeep. Yeah, we have discussed with your VC, so
yes.
That's fine.
Yeah, Shomudip, do not worry. You are already registered. You don't have to worry. You just take the classes, follow the classes, and you'll be just fine. So don't worry.
Yeah.
So any more questions you have? I'm just trying to figure out. No, I don't think there's any more questions. Yes. Okay. So if that's all, then we call it a day. So anybody who wants to join the course, please be a little more initiated and send us email about the payment. For example, Sylvia has asked about it. So maybe Sylvia will send you the details about the payment in dollars so that you can pay totally online. And if you don't have any more questions out here, we can call it a day. So Thank you for listening. And Mohit, please go on.
Thank you everyone, and you can reach out to us by our email addresses as well. So thank you everyone. We will conclude this meeting now and see you.
Hello.
Okay, we have a question, I guess.
Yeah, I was going to type, but I thought because you're saying thank you, I just thought I'll unmute and tell it.
Sure, tell me.
Yeah, this is Sunita, and I had a question. There is on the portal which says the $45 for a course, and what is the 99 membership? that it talks about?
Yeah. So, Sunita,
Sunita, can we take it offline and we discuss?
let me give her an overview. So,
Okay, sure.
so that's the cost of a research license, a membership, which gives you an access for an educational license that we are using it for now. You can upload data for up to 1 MB, and there are educational data sets that are already been uploaded, as Pratim said. But when you're working on your own data, and if you have a large number of data sets to look into, then you would need more space and all those things required and associated with it. So for that, we have a research license that is for $99, and you can have like a lot of more access using that license.
So that also talks about these videos and courses, right? So if I take that, what is the advantage?
Advantage is that you're getting everything. I mean, for $99, you're getting access to everything, and here you have limited access to the educational part of it. When you're doing your own research, on your own project, that is helpful there.
Okay,
So
because
you get an entire cloud space for analyzing your data. For this educational program, we typically give 5 terabytes of space to each person. So for you, might be when you are trying to analyze your own set of data, you might require, let's say 20 terabytes. So that's the research license.
Yeah, and those projects have to be very well defined. So
yes,
yeah,
yes, yeah.
Okay, I think we can talk about it later, little bit
yeah,
later.
Sure,
Absolutely, Sunita. Thanks
I'll
for
go
calling
ahead and
upon
drop
us.
an email about it.
Thank you.
Perfect.
That would be great.
Yeah, thank you so much.
Thank you so much. Yes, yeah.